PCR as an alternative to genomic DNA cloning. We discussed that if we are going to digest the source DNA, it will produce huge number of DNA fragments, and the genomic library that will be produced, it contain variety of recombinants including our gene of interest. So it is a laborious work to screen such a type of genomic library. On the other hand, if we will use polymerase chain reaction or PCR to amplify our gene of interest from a complex genome, then the target gene, it is very easy to isolate if we use specific primers. इसका मतलब ये है कि अगर हम किसी जीन को एम्पलीफाई करना चाह रहे हैं बाय यूजिंग पीसीआर, तो मोर और लेस हमें उसका आइडिया होगा कि इसका जो सीक्वेंस है जीन का वो ये है, उसकी लेंथ इतनी है और इसके लिए ये प्राइमर यूज होंगे. तो इस तरह की प्री कंडीशंस इसके लिए रिक्वायर्ड होती हैं. वंस वी यूज gene specific primers, we can amplify our gene. Then again it depends upon the efficiency of the PCR, that how much length of the DNA it will produce. Comparatively, under standard conditions, PCR can produce 5 kilobase DNA fragment, and for most of the genes this length it is not suitable. And under some circumstances, under standard conditions, around 1 to 2 kilobase DNA fragments, they may be amplified. So for some gene it may be suitable, but for the majority of the gene, this length it is not suitable. So we can say that we need comparatively longer DNA fragments. On the other hand, it also have limitation that the target DNA, it is exposed to high temperature during the denaturation process that can affect the processability of Taq polymerase, because at high temperature some you can say that mismatches may be there, so unwanted bases may be incorporated, nicks may be produced, and Taq polymerase, it is having low proofreading activity. So such type of limitations are there. Now the question is that how we can produce longer DNA fragments. It can be produced by using a long PCR. So in this case, the standard procedure, it can be modified, like the temperature can be lowered, pH can be increased, that increase the stability of the reaction mixture, and even two type of polymerases can be used, like Taq polymerase, and in addition to that PFU thermostable polymerase, it can be used, that has high proofreading activity. So the DNA fragments that we will get, it is appropriate with very low number of you can say that mismatch base pairs or unwanted base pairs. So the DNA fragments that will be produced because of high proofreading activity of the PFU polymerase, it is suitable for further cloning. Now different type of companies, they can provide on commercial basis the mixture of these polymerases that can be used in PCR reaction, so that we can produce longer DNA fragments that may be up to around 20 kilobase pair. Another strategy that can be used to clone PCR product, it is fragment PCR. Because sometime if we want to attempt DNA amplification from single cell or some you can say that complex source of DNA, then very low number of fragments they can be amplified because of the low quantity of the DNA. Then another strategy is that we can use set of primers or random primers that can randomly amplify DNA fragments that can be used for subsequent cloning. So in addition to create genomic DNA library by digesting the source DNA, if we know the target sequence of our gene of interest, it can be amplified specifically by using specific primers, or even randomly by using some other set of primers that can amplify the DNA more randomly, and then these fragments, they can be used to ligate into a suitable cloning vectors for subsequent transformation.